Okay, so in this chapter, okay, first of all, we need to know what enzymes are. Of course, every time we always start with the definition, right? We know that enzymes are the biological catalysts. What does that mean? What is a catalyst? Catalyst is something which has a capability of accelerating the rate of a chemical reaction, okay? But it does not change the equilibrium of the reaction, and the enzyme itself is not changed. That is very, very important. So you can compare an enzyme with your car's accelerator. Okay, as you see, even if there's no accelerator, you can still start the car, right? But the accelerator is going to, if you push the accelerator, it's going to increase the speed of the car, isn't it? So accelerate. So reaction will definitely take place, irrespective whether the enzyme is there or it is not there. Okay, reaction will take place, but it's going to speed up the reaction. Okay, without being consumed, of course, right? So that is very, very important. And with not only without being consumed, without actually changing the equilibrium of the reaction. Maybe this reaction was an exothermic reaction. Because of enzymes it became endothermic. No ways, never ever, never ever. Okay, it's not going to do all that, right? That is very, very important. Okay, so most of the enzymes are proteins, as we know that, except the ribozymes. We recently spoke about the splicing reactions by SNRPs, right? The small nuclear RNA, right? So except that, you can say that most of the enzymes are proteins. So we just said that enzymes can what? They're used as a biocatalyst, right? They can increase the velocity of the chemical reactions. Not all proteins are enzymes, absolutely. All enzymes could be proteins, but of course, not all proteins are the enzymes, right? So all enzymes are proteins with the exception of the ribozyme. That is an exceptional case as in here, right? And then we also have abzymes, okay, the antibody-based enzymes. That is also an exception, okay? They are enzymes which are antibody-based enzymes, okay? Yes, lowering the activation energy. And when it lowers the activation energy, it lowers the activation hill. Okay, we'll talk about that maybe, after a while. So without the enzyme, the hill will look like this. Okay, and with the enzyme, the activation energy is going to be like this. So which one is, easier to climb and move over? The first one or the second one? Which one will take less time to move on? Obviously the second time, right? So with the help of enzymes, okay, this would be the activation energy, right, for reaction. So it will be easier to actually cross this smaller hill than this taller one, right? And as I said that, for example, if you choose to go from your home to the Al Faisal University by walking, anyways, you will reach to the Al Faisal University, you will reach to the university, right? So, but if you choose to go in your car, or if you use a chopper, right, you are going to reach pretty much earlier to the university. Okay, it's the same thing. Anyways, the reaction will take place, but with the help of enzymes, there will be the velocity of the reaction will increase. The reaction will move faster. It will accelerate, right? So that is the difference when we use the enzymes. Okay, because what happens, we'll talk about that in a while. Enzymes, they provide a special environment to the reactants. If I have A plus B, okay, which is going to be converted into C as a product, if I choose to use an enzyme here as a catalyst, this enzyme is going to provide a special place, a special environment, okay, where these chemicals will be modified. Okay, and these chemicals will be modified, and they will be modified in such a way that it can easily, A plus B will be easily converted into the product. Okay, so, and that environment is created by the enzymes. Okay, and least amount of energy will be required to convert A plus B into C when we use the enzyme. Very less energy will be required. Okay, that is called as that is the basically effect of the enzymes. Okay, because you will see that in a while, since these enzymes are made up of proteins, and these proteins have certain special amino acids. Okay, and these amino acids are so special that they provide, a kind of, ideal environment, or binding site where A plus B can easily, you know react with each other. Maybe, the amino acids in these enzymes, they bring these A and B molecules together, you never know, right? Thus creating an environment so that the reaction can speed up. Okay, so that the reaction can speed up. Okay, and now, these enzymes are very, very specific. When we talk about the properties of the enzymes, okay, uh, we will also talk about, at certain places, we need to use coenzymes, cofactors. I'll explain what is a coenzyme, what is a cofactor. I'll explain you all that, right? But first of all, these enzymes are very, very specific. When I say specific, what does that mean? That means they cannot catalyze any reaction in the world. No, right? For example, I have an enzyme called the urease. It is a very, very specific. Why is it specific? Its specificity lies that it can act on just a substrate called as a urea, okay, and then convert this urea into the ammonia. That is its specificity, right? You cannot expect the urease can act on every substrate. No, right? It has some specificity. Okay, number one. Or they're stereoselective. What do you mean by stereoselective? Some enzymes, they act only on L amino acids. Like there's an racemase. Alanine racemase is an enzyme that acts only on alanine. That is it, right? So that means they are stereospecific. They're they show optical activity. Their optical specificity, right? That is very, very important. And some enzymes show geometrical specificity. That means they can act only on cis isomers, not on trans isomers, right? There are certain enzymes, which show geometrical specificity as well. Okay. And then, of course, we all know that, being the anatomist, since we study anatomy, we all know that what are proteolytic enzymes. Proteolytic enzymes are those enzymes which can catalyze what? The peptide bond, isn't it? Like, the enzymes secreted by our pancreas, right? The pepsin, trypsin, chymotrypsin. We're going to talk about all that in digestive system, right? They split the polypeptide, they split the peptide bond. So that means they are specific in breaking the peptide bond. They cannot break any bond in this world. They can break only the peptide bond. So this was just about the introduction of the proteins. Okay, sorry, enzymes. Name an enzyme which is not a protein due to its active site. Yes, Faisal. We'll go one by one, Faisal. You're right. We'll go one by one. We'll discuss everything in detail. Don't worry about that, Faisal. You're absolutely right. They have an active site. They have an active site, specific portion in the enzyme, okay, to which the substrate can actually bind. That is called an active site. You're right. Not all. We'll not go into exceptions. But who will answer this question? Name an enzyme which is not a protein. Yes, ribozymes, isn't it? Absolutely, ribozymes. They are not proteins, they are RNA molecules. Absolutely, the SNRPs. Where are enzymes synthesized? Can we ask this basic question to ourselves? Where are enzymes produced? Do we know that? Did you study that in anatomy? And by which process they are produced? How do we produce them? RNA, okay. Where are they produced? Ribosomes. Absolutely. You know, Sometimes there are very simple questions, and we take a lot of time to answer that, because we're like, oh my God, this is so simple. Yes, sometimes we do ask very simple questions. Enzymes are formed at on the ribosomes, okay, and those ribosomes are attached to the rough endoplasmic reticulum, because they are proteins. Because in order to answer this question, you know that enzymes are proteins. So, of course, they are made in the ribosomes. Now the classification of the enzymes. Okay, now let me tell you what you need to know about this, because I've also received plenty of questions about this. What do we need to what all we need to memorize in this? Okay, so first of all, we have to know all the classes of the enzymes. This is basically the classification of the enzymes. Okay, enzymes are classified into six major groups. Okay, according to the type of reaction they catalyze. Okay, and like oxidoreductases. Now, what do you need to know? Now, please pay attention. Nobody is going to ask you, give me an example about oxidoreductases, like how pyruvate is converted into lactate. No, of course not. Okay. You might be asked, for example, you don't have to know the reactions as well. You might be asked a question like lactate dehydrogenase is an example of what kind of enzyme? Number one, a question might be like this, so you have to answer. It's an oxidoreductase enzyme. Khalas. Nobody's going to ask you what is converted by lactate dehydrogenase, and no structures, nothing. And second thing, what else you need to know, that someone will ask, the question might be, for example, about oxidoreductases. What is their function? What do they do? They catalyze the oxidation reduction reactions. Khalas. That's all you need to know. You need to know the name of six classes of the enzymes. You need to know one example from your slide, just the example. Number two. And number three, you need to know, for example, if I say oxidoreductases, what does this enzyme do actually? Umar tried hard learning international languages. Oh my God, this is so good. I am going to copy this and I'm going to send it to everyone on Moodle. Thank you, Mohammed, for that. Very good. Thank you. Thank you for sharing that. I'm going to share that with everybody on Moodle. Guys, he has a very good mnemonic, Umar tried hard learning international languages. Right? Definitely, I'm going to share that. Thank you, Mohammed, for that. Thank you. Okay, so by using this, so he has already given us a mnemonic, right? So you can remember this mnemonic and you can remember the enzymes. Okay, and I'm going to put this in my presentation and use it for the next year students as well. Okay, so a question might be, so what does your oxidoreductases do? You have to simply say that they catalyze those reactions where in the oxidation and reduction of the substrate will take place. Khalas, that's all you need to know. Is it clear, everyone? One example, just the name of the example. Okay, and number two, you have to know what the name of the enzymes, and number three, what exactly is the role of the enzyme in a particular reaction. Okay, so now transferases. What do transferases do? As the name itself says, that they transfer the functional groups between the substrates. Remember, look at this. I had this functional group over here. Okay, and then look at this. I have now this functional group over here. It was transferred from one reactant to the other reactant. That is all you need to remember. Example, aspartate aminotransferase. What kind of enzyme is it? It's a transferase enzyme. That's all you will remember. Clear, guys? No confusion? What you need to know about it, because this always, students get scared by these structures. So that's why I try to let them know what exactly they need to know. Okay, moving on further, hydrolases. Hydrolase, as the name itself says, whenever there is any hydrolysis reaction, okay, wherein simply the bond is broken or cleaved with the what do we use? We use water here, isn't it? For hydrolysis reactions, we always use water, isn't it? Water as a functional group. Okay, we break the bond and then add water to wherever there is, right? Yes, so hydrolysis we use water. Okay, as you can see over here, I used water over here. Okay, and look at this, that OH was added to the choline right over here. Okay, so this is kind of reaction is the hydro. What kind of enzyme is an acetylcholine esterase? It is a hydrolase enzyme. Liases. What do liases? So liase is something, right, where I have seen that students, what do? They think that liase is an enzyme which joins together, because That is a different, that's a ligase enzyme. So please do not mix lyase with the ligase. Ligase is different, lyase is different. So please be careful with these two words. Lyase and ligase is, they're totally different. Okay. Lyase is an enzyme, okay, wherein two groups are added to a double bond. Okay, if I have a double bond here, so we add whatever groups there are. As I told you, you just need to remember, what exactly happens with the lyases, right? So we add groups to the double bond, or sometimes remove the groups from the double bond, right? So that is basically done by the lyases. As you can see over here, there was a double bond over here. Look at this, there was a double bond over here, right? So by simply adding here what H and OH, H was already there, OH over here. What did we do? We removed the double bond by using a lyase. So lyase are basically specialized in doing what? In creating a double bond or in removing a double bond, in breaking a double bond. Okay, that is all you need to know about it here, about lyase. So what kind of, you need to know the examples. Aconitase is a type of what? Lyase enzyme. Okay, that's how you will remember. Then you have isomerase, the most easiest of all. Of course, it is going to, guys, wait a minute, let me charge my laptop. Charge is going to get over in a minute. I've been using it since morning. I think, yeah, we're okay. Yeah. Okay. So what do we have? Then we have the isomerases. Isomerase is something like which is very, very important, right? And which is very easy to memorize, very easy to remember, right? So isomerases are, they're those enzymes which, which are involved in isomerization reaction, converting cis into trans, trans into cis, right? So that's what it is. And the example here, it is the phosphohexo isomerase. Okay. So that's what you need to know. And then we have the ligases here. Ligase and lysis are different now. Ligase is, joining two things together. That is a ligase. Okay. So ligase are those enzymes when you have to, join the two substrates. Okay. And we usually use ATP in these kind of enzymes, right? If you see here, we use ATP in these kind of enzymes, okay, in the ligases to provide the energy for joining. Okay. So that's all about the six classes of the enzymes. Is it clear? You have the mnemonic now. You know what is expected out of you. Okay. All clear, no confusion. Should I move to the next topic? Okay. And then you will just read this slide by yourself. Blood, what kind of enzyme is present? We have thrombin, okay, which converts fibrinogen into fibrin, right? Your pancreas, they secrete a lot of digestive enzymes which cause hydrolysis of the proteins. Mitochondria, whatever, the citric acid cycle we're going to talk about. There's so many enzymes which are present in the mitochondrial membrane. Okay. uh, you know this we're going to talk about in the last chapter, in the second last chapter. Lysozyme, recently we spoke about this, lysozyme, okay, which can cause, if you remember, where do we have lysozyme? First line, second line, third line of defense mechanism in
First
lymphatic
line,
system. First line. Okay, where do?
NRTs,
Tears.
lacrimal
Very good, absolutely. In tears as well as in the saliva as well, right?
saliva. Yeah,
Yes, very good, right? That's how you can connect the two subjects. So protease, so you just lipases, we know lipases are, but don't forget, you have to memorize this table as well. So don't think that I did not teach you in detail this one, so we don't have, know this. You have to know what are lipases. What do lipases do? They split the fat. You already know all this, right? Amylases for carbohydrates, nucleases to split the nucleic acid. If you remember, recently we spoke about exonuclease and endonuclease. Remember when we were doing BER, right? When we were how to repair if there is when there's a proofreading, how to repair, if there is any mistake done by the DNA polymerase, right? So we said about exonuclease and endonucleases. So nucleases are the enzymes which splits the nucleic acids. Okay. So proteases and peptidases to split the proteins. So please study these three tables as well. They're very easy to memorize. Okay, don't ignore them. Okay, and then about acetylcholine. Okay, we know that acetylcholine is a what? What is acetylcholine? It is a neurotransmitter. What is it known for? We learned that in last chapter. On Yes, we need it for the, remember the neuromuscular, I should have put the diagram here. Remember the neuromuscular junction? We had a diagram something like, where we had the end of the axon terminal, a diagram which looked like this, and then here we had a sarcolemma, right? And this was a synaptic cleft, and here we had the receptors for the acetylcholine. And if you remember, we discussed when the action potential reaches here, the calcium goes out, right? The calcium moves, there's an influx of calcium. Then we have acetylcholine, okay, in these synaptic, vesicles, which is released, which binds to its receptors, and that's how the channels are open, right? We discussed that. And if you remember, how, how do all these things end when the muscle has contracted? Now we want muscle to relax. What do we need to muscle to relax at the neuromuscular junction? What must be degraded to stop the muscle to make the muscle relax now? We need an enzyme. If you remember acetylcholine esterase. Acetylcholine esterase was an enzyme. If you remember, we used to write it like this: ACHE. Remember, I told you that enzyme is always present in the synaptic cleft. Okay, so once the action potential is over and the muscle needs to relax, then we need an acetylcholine esterase, okay, which can actually hydrolyze acetylcholine into, we did that last semester, into acetic acid and choline. Maybe if you don't remember, because we didn't do the structures, we just did it orally, like acetylcholine is degraded into acetic acid and choline. And if you remember, I told you they are basically recycled then, reused then once again. Yes, absolutely. And this is the same thing. They have given an example of acetylcholine esterase. It's also an enzyme found in the synaptic cleft, converting acetylcholine into acetic acid and choline. Okay, that's all you need to remember. And then succinyl choline. You might have not heard about the succinyl choline. Okay, this enzyme is called as a neuromuscular blocker. Okay, why so? Because whenever we need to paralyze a particular patient, okay, and now you might think that why do we need to paralyze a patient? Because for example, if you're going to operate upon a patient, we are going to do a surgery, okay, or you might do a bronchoscopy, okay, or the person is put at ventilator, right? So at that time you might need to paralyze the muscle of the patient, okay. You might need to you might want the muscles of the patient should be in a relaxed mode, okay, especially when you need to do intubation in the trachea, okay. So in order to do the intubation, okay, you want, or you want to do a dental surgery, okay, you in the intubation you want the patient's mouth, okay, or patient's vocal cords, okay, or patient's jaw should be relaxed, okay, so that he doesn't move, right? So at that time you use succinyl choline, okay. So at that time there's a competition between acetylcholine and the succinyl choline. It's the succinyl choline which will bind to the receptors over here, okay. Are you able to understand, guys, what I'm trying to say? Okay. And then later on the succinyl choline can be it can be hydrolyzed by the acetylcholine esterase. But this succinyl choline is hydrolyzed very slowly by the acetylcholine esterase. So that is why you can keep the patient's muscle relaxed for a longer time, okay. So this is usually used whenever you have to give anesthesia to patients for the short duration, like its life is basically for 8 to 10 minutes, or maybe sometimes 15 minutes, okay. We call it a neuromuscular blocker, okay. So that was about the excellent use of an of an enzyme. Okay, these surgeries or this anesthesia is possible only because of the enzyme, okay, available here. That's what it's trying to explain you here. Okay, and then you have certain these definitions which we'll talk about in a while. What is an active site? What is an active site? You need to know. Yes, guys. So please know all the definitions. Please, everyone, know all the definitions very carefully. Don't think that, okay, we don't need to know the definitions. No, it's not like that. You need to know the definitions as well. Okay. So what is an active site? Active site, you can say that, a pocket or a specific portion, or, a part in the enzyme, okay, where actually the substrate binds. Okay. So if this is my enzyme, okay, I might be having a active site. This is the place where my, let me get another color. This is the place where my substrate is going to bind. This is called as an active site. Okay, so it's, you can say that it's like a pocket in the enzyme, okay, where the basically where the substrate binds to it. Okay, substrate binding site is an active site. So what is a substrate? Substrate is basically that compound, okay, which is basically, where the enzyme will act on. Okay, so substrate is the one which is going to bind to the enzyme, okay, and form the enzyme substrate complex. So I have an enzyme, then I had a substrate, both of them joined to each other, and it formed the enzyme substrate complex, which was an intermediate. Later on, my enzyme was released, and then it resulted in the formation of the product. Okay, so substrate is equally important. Okay, anybody? Gada. No, the difference is, Gada, you're right. The difference, acetylcholine is naturally found in our neuromuscular junctions. Succinyl choline is, which you're using artificially. The difference is acetylcholine esterase is going to quickly break the acetylcholine into acetic acid and choline, while as it's going to take longer time to break the succinyl choline. So that is why giving you that extra time when the muscles can be relaxed. Is it clear, Gada? One is the natural one, another one is used as a medication, as an injectable given to the patient. Okay. Okay. So now I'm going to explain this in a while. What is an apoenzyme, cofactor coenzyme? I'm going to explain this in a while. Okay. Let's first of all try to see the active site. Okay. Look at this. This is, oh, where's my pen? This is an active site. This is an active site. Okay. Where the substrate is going to bind. Now, very important, everybody pay attention. There are certain terms which you need to know. What is the difference between a hollow enzyme? What is a hollow enzyme? So please pay attention, everyone. What is the difference between hollow enzyme? Okay. And then you have to remember that there's something called as hollow enzyme means whole. Hollow means everything. Kullu, right? So hollow enzyme is something, okay, which is actually an active enzyme, which is a protein part of the enzyme. Hollow enzyme, okay. This hollow enzyme is the one, okay, which is made up of two things. Hollow enzyme is an enzyme as a whole, which is made up of two things. So please pay attention. It is made up of, okay, apoenzyme. So please remember this classification. It's made up of an apoenzyme. So what is an apoenzyme? Apoenzyme is the protein part of this hollow enzyme. So apoenzyme is the protein part of this enzyme. Now the confusing part. Okay. Yes, Apo is an active enzyme, right? The whole enzyme, you can say.